Hello, this is Mahendra Didia. This is module number 6 and it is part of the book on liquid formulations. This is the second related video submodule focusing on auxiliary agents. In previous submodule, we looked at solvent, co-solvent and surfactants. The auxiliary agents include solutes as well as solvents used in formulation to provide its effectiveness. Buffering agent. Buffering agents are used to stabilize the product. pH can be acidic or basic depending on formulation, and generally buffers are recommended to resist this change in pH upon addition of acid or base or during storage. Buffer is a mixture of weak acid and its conjugate base and vice versa, that is weak base and its conjugate acid. Buffer also stabilizes the solubility of the product. It maintains the pH and thereby the solution of the solute. Example, buffering action of an amino acid. Amino acids are both groups, amino group and the acid group. When we add HCl to that, it donates its hydrogen to the amino group and the pH remains constant. When we add NaOH, sodium ion reacts with COOH, forming a sodium compound, and it keeps the pH constant. So the buffering agents allow pH to remain constant and thereby provides its effectiveness. Buffering agents used in pharmaceutical and cosmetic formulations. These are alphabetically listed, and the pH ranges also is included a product in which these buffers are used. This is just an example. Acetate salt provide a wide range of buffers depending on the pH of the product. Arginine is used, basic pH 7 to 7.4. Boric acids and borate salts used in ophthalmic solutions. Citrate is very commonly used in both cosmetic and pharmaceuticals. Most common is sodium citrate. Citric acid combination has a wide range of 3 to 8.5. Biotechnology products use histidine hydrochloride combination with pH range of 5 to 6.5. This chart continues. Lysine is also used. Phosphates pH configurations and buffer configuration, and they are used for different pH controls, different buffer conditions. Solubility, pH and buffer. Here's an example of propylparaben, commonly used preservative in pharmaceutical products. pH range from 2 to 8.5. Propylparabens more soluble in basic pH, ionized in this pH. In order to have preservative effectiveness, propylparaben needs to be in unionized form, anywhere from pH of about 3 to 7.5. Drug sulfadimethoxam, equilibrium pH with different buffers are shown here. Buffers: phosphate, formate, citrate, acetate, lactate, ammonium, and borate. This compound shows a U-shaped solubility curve with low solubility between pH 3 and 5. Beside the buffer composition that would provide optimum formulation. Preservatives are used in an aqueous solution. Aqueous solution may become microbiologically contaminated due to raw materials. They provide growth media for bacteria and molds such as water, gums, and sugar. Sugars are also used to provide sweetness. Gums are used to provide viscosity and thickness. And introduction of microorganisms can also happen during manufacturing and consumer use. Oral solutions are opened several times and consumer would withdraw small samples for administration. This could introduce microbial contamination. So preservatives are used to protect formulation from microbial growth. It's recommended that formulator be aware of the microbial testing and how to interpret the results. In this video, a portion of testing is shown. So antimicrobial efficacy testing is required. Microorganisms are used. Several microorganisms are tested. Growth plates are used, such as shown here. Microbiological inoculum is added to the solution. Preservative in the solution should be able to prevent from microbiological growth. Requirements of United States Pharmacopoeia is used to determine the microbial effectiveness of the selected preservatives. Preservative characteristics be able to prevent the growth of microorganisms. Selection of preservative is alone or in combination. For example, parabens are used in a combination, whereas benzoic acid will be used alone. It is designed to prevent growth of microorganisms. It should be effective over a wide spectrum of microorganisms. Gram-positive, gram-negative, more than fungi, yeast specified in United States Pharmacopoeia. In addition to that, preservatives used should be stable at effective concentration throughout the product shelf life. For example, methyl and propylparabens are effective at pHs 3 to 7. They need to be non-toxic and non-synthesizing. Preservative needs to be compatible with ingredients used in the dosage, convert into forms where they would not be effective. Preservatives are solute. That is, solubility of all solutes, including preservative, is required to prepare a solution dosage form. In addition to that, pH should be such that unionized species of preservative is present. Examples of preservatives. Very commonly used in pharmaceutical, and it has been approved by FDA. It's listed in USP. However, in cosmetic products, there's a reluctance to use based on articles published in magazines and journals, which are not supported by FDA. Pharmaceutical industry continues to use parabens, and there are no reported issues. Concentration up to 0.2% are used. pH generally is 4 to 8. Generally, 2 esters, methylpropyl or methylbutyl, will be used to achieve the activity against broad spectrum of microorganisms. Quaternary ammonium compound, benzalkonium chloride, is commonly used in ophthalmic solutions. Benzoic acid and sorbic acids are commonly used in pharmaceutical preparation as well as some cosmetic preparation. Ethanol and propylene glycol, if that concentrations are high, for example 20% weight to volume, also becomes a preservative. Antioxidant chelating agent. Antioxidants are used in pharmaceutical as well as in cosmetic products. Antioxidants react with free radical. Example is ascorbic acid. Ascorbic acid forms an ascorbate. Ascorbate captures the free radical shown here as a dot. This will then convert into dehydroascorbate. So ascorbic acid acts as an antioxidant by capturing free. Commonly used are vitamin E and its derivatives. Sodium bisulfites are also commonly used in pharmaceutical. Chelating agents are used, such as EDTA, to chelate metal ions. So metal ions are trapped and prevents reaction of that metal ion with the drug or active ingredient. Rheological agents, in addition to solvents, co-solvent, pH, surfactants, preservatives, rheological agents are extremely important. Oral solutions, injectables, dermatological solutions, cosmetic solutions have pourability component. And pourability should not be runny or gel-like where it's unpourable. That requires adjustment of rheology, viscosity, and density of the solution. So we can accurately dose the product using different dosing devices. Rheological modifier in liquid dosage forms are cellulosic derivatives, gum, for example, xanthan gum, and several synthetic polymers, carbomers, PVP, PVA, and polaxamar are some of the examples. Shown in this table is density and viscosity of acetaminophen solution, cherry flavored 5 ml, and the measured dose and its accuracy in different devices that are used, such as dropper, syringe, and cups. And accuracy needs to be established for different devices. For example, for 5 ml, droppers provide 4.96 mean value. Syringes are showing slightly higher, so that syringe needs to be recalibrated. Are showing slightly lower, so this cup needs to be recalibrated. This type of study helps in designing, in this case, syringe and cup. Taste, color, and fragrance. Many drugs and solutes have unpleasant or bitter taste and requires taste masking with sweeteners, flavors, and FDA-approved color. This is a requirement for palatability, and this is one of the quality by design criteria. FDA will look at palatability of oral solutions. Sweetening agents, sucrose is commonly used, sorbitol is also commonly used, mannitol, glycerol, saccharins not used widely, aspartame, acesulfam K, sucralose, and there are several sweeteners that are used in formulation. Flavors are used to mask unpleasant odor and also enhance the taste, because flavor and taste go together. They're natural and artificial flavors used. Some of the common ones are peppermint, lemon, grapes, etc. Colors, both for cosmetic and food. They are FDA approved colors. They are either dyes or lakes. Most of them are known as artificial or certified colors and can be used in wide combination. Formulators should always look at the FDA guidelines for both cosmetic and pharmaceutical. Here are the examples of colors range from blue, green, orange, red, violet colors, and yellow. Some of these colors will require declaration on the label. For example, yellow number 5. Look at the colors and the guidances that are offered by FDA and worldwide regulatory agencies. Not all the colors are approved by all the agencies. A color may be approved in US by FDA, but it may not be approved by EMA in Europe. In addition to solvents, co-solvents, surfactant, auxiliary agents are included in formulation to make formulation usable